Next, we're going to look at chemical risks. Toxicology is the study of chemical risks. And there are 6 categories of chemical risks that you will learn about. The first are neurotoxins. These are chemicals that disrupt the nervous system. Think of the word neurology as a study of the nervous system, or if you took anatomy, you might know the word neuron or nerve. So these neurotoxins are typically found in insecticides that can affect all kinds of organisms, not just human, but we are talking about human risk here. Lead, which you've heard of before, it's a heavy metal, and it affects the development of the brain and nervous system, particularly in children. And mercury, which is another heavy metal that also disrupts the nervous system. In this case, adults are as affected as children. If you'd like to know more about the effect of mercury, there's a case study that I've posted. It's about a 5-minute video about a case in Japan, a place known as Minamata Bay, where a nitrogen fertilizer plant released tremendous amounts of heavy mercury, and you can see the devastating results that this had. And it's another example of environmental justice or injustice, because approximately 10,000 people were affected, and it took over a decade for, even though they knew pretty quickly, within about 5 years, they knew what the cause of this disease was, it still took another 10 years after that for this mercury dumping to stop. Okay, a second category of chemical risks are carcinogens. These are chemicals that cause cancer by inflicting damage to cells. Carcinogens include tobacco smoke, which inflicts damage to lung cells, asbestos, which is a mineral fiber that was found in floor tiles and ceiling tiles, and other products, and also causes lung cancer. And radon, which is a gas that may be present in a foundation like the basement of a home and coming through the home. So if you're interested in learning more about radon, I also have attached a little flyer where you can learn about how radon can be tested for and how it can be blocked from coming into your home, as well as the damage that it causes. Okay, the next category of chemical risk are teratogens. Teratogens are chemicals that interfere with the normal development of embryos or fetuses. So a teratogen can include, 2 examples are alcohol and certain medications. Back in the 1950s, there was a medication that was given to women who were pregnant. It's called thalidomide. And this chemical caused the fetuses to develop or not develop limbs properly. And it was had never been approved by the FDA at the time. It had been approved by the Canadian equivalent. However, the FDA also had information that they sat on for a very long time. They didn't act on information that was coming in, which indicated that thalidomide actually did a lot of damages. So they really shouldn't take any credit for that sitting on things, but after that, drugs were moved to more fast-track development. Okay, and then alcohol, of course, if someone drinks alcohol during pregnancy, it will affect the brains of the fetuses. 2 others, one is a subcategory. 2 other types of chemical risk are allergens. Allergens, which are chemicals that cause allergic reactions. The reaction can be minor or it can be very serious and even fatal. It's now known that people can have allergies to certain nuts, even certain fruits, penicillin and other medications, and not typically serious, but also bothersome, animal dander. And so if anyone can develop an allergic reaction or an allergy to something at any point in time. So just because you didn't have an allergy previously doesn't mean that you won't develop an allergy. Another example of allergens causing serious illness are the nurses that I told you about who were exposed to latex in Massachusetts hospitals. And as they removed the latex gloves that they were using while working, little particles of latex flew into the air and eventually caused an allergy to the latex. In addition to that, certain cleaning products that were being sprayed with spray bottles to clean, let's say, the operating room bed were also causing chemical sensitivities or allergies, some of which became so severe that these nurses could no longer work, and some even had issues with doing things like walking down an aisle in a grocery store where various products, cleaning products with certain chemicals were in it because their immune system began to react to all sorts of chemicals, not just the latex. Okay, last and not least are endocrine disruptors. These are chemicals that interfere with the normal functioning of hormones in an animal's body. So first of all, what is a hormone? A hormone that is a chemical that is produced in one area of the body, one organ in the body, and then acts on another organ of the body. Some hormones or examples of hormones that you might have heard of, for example, are thyroid hormone, which is produced in the thyroid but then acts on the metabolism, and reproductive hormones, which act on other organs. And one category is the hormones that are given to livestock to increase milk production, for example. And these hormones may affect humans when they take in milk that has been treated with hormones. Another issue is that there are a number of chemicals that mimic hormones, natural human hormones. And when they mimic them and they get into the human body, they then interfere with the proper production of or action of natural hormones. So this leads to a subcategory of chemical risk, which are hormonal wastes. Hormonal wastes are wastes that pass through, often through humans or let's say cattle, and then affect fish and amphibian reproduction, and may also affect humans. Okay, now we're going to look at how scientists determine at what concentrations of the chemical would be harmful to organisms and to human health as well. And, excuse me. There are studies called dose-response studies. You increase the dose over time, excuse me, and you look at the response of the individuals. So the first one is a lethal dose 50. That means the concentration at which 50% of the tested individuals die. So perhaps the test individual would be a mouse. So then if you increase the dose of a chemical that the mouse is exposed to, the point at which 50% of those mice die is called the lethal dose 50. We'll look at that further on the next slide, but first, it just know two facts. It determines a relative toxicity of a chemical. It's not an absolute, and it varies greatly among species for any given chemical. So what is harmful to a mouse may be even more harmful to a fish or an amphibian. So that's something to keep in mind. Sometimes there are harmful effects that aren't deadly, not leading to death. And those are studied with a measurement called effective dose 50. Effective does not mean in a positive way. It doesn't mean, the dose at which a medication, for example, is helpful. Effective dose is the dose that causes 50% of, ED50 that is the dose at which 50% of the animals tested display a harmful but non-lethal effect. So it might be an effect on their ability to reproduce, for example. It might be an effect on their behavior. So it's an effective dose on 50% of those test organisms. Now let's look up here at a typical hypothetical graph. We have the doses increasing from, in hypothetical units, 0, 2, 4, 6, 8, 10. And on this axis, we have the percent mortality, or the number of test organisms who die. So you can see that the curve is one of those S curves. It's flat for a while, then an exponential increase, and then a slowing down of the curve to the point where the remaining organisms survive or don't. So you would look across, or a scientist would look across at the 50% mortality rate, follow it across till it intersects the dose, and then follow it down to the x-axis and determine the lethal dose 50. So in this case, the lethal dose would be 5.5 hypothetical units. So let's take a moment to consider how we translate that into what limits are placed on these chemicals. So the rule of thumb for chemicals that are allowed to be released into the environment is to take the lethal dose 50 and divide it by 10 and say that's a safe dose. So if you have a lethal dose 50 of 5.5 and you divide that by 10, you'd get 0.55, and that would bring you down to about here on the graph. And that point on the graph, there appears to be a very low risk. For exposure for humans, we take an even greater safety and divide it by typically 1,000, and that would bring us down to, 0.0055, which would bring us down all the way to here. Sometimes they divide only by 100, but 100 or 1,000. And so we divide and we get down to this point in the graph. So that you can see how those numbers are derived. They're derived from a graph like this, saying that makes your risk very low, but it doesn't make your risk zero, because it's not really looking really in depth at what happens at that dose. It's sort of a magic number pulled out of a hat from common sense. Now, most of the studies, dose response studies that are done, are these things called acute studies. They're short term. They last only a few days, and they typically involve highish doses of a chemical. Chronic studies are also important, and that's the attempt to observe the effects of long-term exposure to a chemical, sometimes at low dosage. So we might start with an acute study like this, but then move to chronic studies where we attempt to study chemicals over a long period or repeated period of exposures to a lower dose. I'm going to pause here and move on to a new screencast.